talking about in-process monitoring tools in quantitative RT-PCR and how they can help to improve the accuracy and reproducibility of your results. My name is Dirk Schacht. I'm working at Product Management in Hilden with QIAGEN. Before starting the session, please let me quickly note that when talking about products, or mention that when talking about products, these are not meant for diagnostic purposes, but for research use. So during our session today, we will first summarize a few topics about controls, why they are used in reverse transcription quantitative PCR when doing gene expression analysis. I will elaborate then a little bit more on internal controls specifically, followed by a short chapter about introducing some additional safety or control measures that can be incorporated into qPCR procedures. And finally, I will show a few performance data about our new QuantiNova kits that I'm going to mention during this session. And of course, in the end, we have the opportunity to discuss some questions and for me to answer them to you and collect your feedback. So let me please elaborate and start with talking about controls in quantitative RT-PCR. So why are we including controls? There are basically a few topics. Key purpose that want to be achieved. You want to feel safe and confident about the work and the results you are doing. You are actually even going to share with others or going to publish, and you are basically looking into relevant insight of your biological samples and the questions associated with them. And for that reason, we are incorporating controls into the procedures to improve the standardization of our process to make sure the accuracy and reproducibility of the experiments is great, to confirm the validity of what we are doing, to reduce any type of variations or variables that would result potentially in artifacts of our experiments, to increase definitely the predictive value of what we are doing, and also to be in line and synchronized with guidelines like good laboratory practice, or I've been specifically mentioning here the MIKE guidelines. If you are interested to get experts' recommendations on how to work with controls, how to monitor quantitative PCR or RT-PCR procedures. This is one of the publications that is a very comprehensive and great overview on how to monitor and make sure that the results we are reporting from quantitative PCR experiments are reproducible and meaningful. So let me quickly talk about controls which are commonly used in reverse transcription quantitative PCR. They are all used or required for different purposes or serve different purposes here. So one thing that definitely should be included in each PCR experiment is a no template control. The purpose of a no template control is to confirm that a positive result really is positive result and not accidentally being caused by any type of contamination. A second thing which commonly is used when working in qPCR with RNA or messenger RNA detection is a no reverse transcriptase control or a control detecting genomic DNA contamination. I will elaborate a little bit more on this topic a bit later on. The key purpose of this is to confirm that a signal we are getting is really specific, caused by the transcript we are looking for. Because in theory and as well in practice, genomic DNA, which potentially is carried over into our purified RNA or into our experiment, will also bind to the primers, or primers can bind to the gDNA, and then cause an additional elevated signal there, which will simply add to our specific signal from the transcript. Another control that was more commonly used in the back is something like a no amplification control. This is not so frequently done today anymore, but it was used frequently, specifically when working with dual labeled probes. If there should be any type of degradation or damage of them, it would increase the background noise and can be ruled out by a no template control or detected by that. Another thing that makes a lot of sense actually is a positive control or an internal control. These are slightly two different things. I will talk a little bit more about that. The purpose of an internal control or positive control is to confirm that the reagents and equipment we are using are really properly working. What it does is actually it will detect any type of either inhibition or malfunction of equipment, for example. But particularly inhibitors play a major role in changing or altering our quantitative results, particularly in gene expression research or experiment. A positive control or an internal control cannot really be replaced by a reference gene or by a housekeeper. These are used for a different purpose. By that they are not really controls, simply because they will not be able to detect any type of inhibition, because also housekeepers or reference genes will undergo a certain variation caused by the efficiency of our RNA, even DNA extraction. Their purpose is to be utilized in relative or sometimes even absolute quantification, depending on which type of experimental setup we are looking at. So let's look a little bit more specific into the internal control, which I already mentioned. It is made to detect contamination by inhibitors in our typically RNA or DNA. So what the internal control overall does, it really ensures that our quantification results are precise and consistent, but it also ensures or shows that the results from sample to sample or from day to day are consistent. So here is a list of what really can influence our quantitation and the precision of our quantification in qRT-PCR. And when thinking about that, it's almost scary at how many different points our experiment could be influenced, or in worst case even be spoiled. So most commonly, or the largest problem certainly, is any type of inhibition, particularly when looking at RNA. And inhibitions can come from either the sample material itself. There are sample types that are notoriously difficult to extract and to handle, but also when utilizing an extraction method that is sufficiently harsh to get access to the nucleic acid we want to view, we incur a high risk of adding any inhibitor by the reagents we are using. For example, when thinking about phenol, this is a very strong inhibitor of proteins. Basically, it's the job of phenol to destroy proteins and denature them. And if we have any carryover of phenol into our nucleic acid, then it can potentially interfere, for example, with our enzymes, with our Taq polymerase or reverse transcription, which are proteins. And it may not always kill the enzyme completely, but it may just reduce the effectivity or the efficiency of the enzyme proportionally. And this is one of the worst things that can happen to a quantitative experiment, because a slight shift of CQ or CT may create even more problems to us than a complete failure of our experiment. Because a complete failure is something that we see. If we are getting no results, we see something went wrong and we will potentially repeat or fix what we have done. But if our CQ value is just delayed by 3, 4, 5 CT cycles, we won't probably even realize that, but we would misinterpret this by a change in the expression profile of our target messenger. And that's critical or difficult to detect at all. Other things that of course could happen to us is the instrument is not working right, the chemistry has been damaged maybe by improper storage situation, or we just do an error when setting up or adding our reagent. So there are various different reasons why we could potentially go wrong, but certainly inhibitors are some of the key issues we need to deal with, and internal control can rule them out. So on this slide we show how that basically is done. And internal control supports the precision of our results or confirms the results by being spiked into our sample at a defined concentration or amount. So that basically means we always have the identical outcome from an internal control. It should have a very, very similar CT or CQ value from run to run to run. And this is how it differs from an housekeeper or reference gene that would differ from extraction to extraction. An internal control shall not differ from extraction to extraction. And it will always undergo the absolutely identical treatment as our target messenger RNA we're looking at. And by this, it will always immediately reveal any kind of inherent issue that happens to our reaction to our PCR tube, essentially. In the best situation, we may use probe-based detection procedures, and then we can even duplex our PCR and use the internal control or run the internal control in exactly the same well as we run our target RNA. So also the internal control always undergoes both the reverse transcription and the qPCR reaction, so it monitors both the reverse transcriptase and the Taq polymerase. So this will result, as I mentioned, in reproducible CQ values if we use the internal control in the same type of experimental setup. And by these results, we will always be sure that we have a reproducibility from assay to assay. QIAGEN has included now the internal control RNA and artificial transcript into our QuantiNova kits for quantitative RNA detection, and our recommendation is we are saying if the internal control stays within 2 cycles around your or within 2 cycles identical from run to run, then this run can probably be considered as being okay. But if the deviation is delayed by more than 2 cycles, then something is probably strange or wrong with this specific PCR reaction or tube, and we should have a closer look to rule out any errors or issues. So the key thing is that the internal control will confirm that our results are proper and comparable. Here are a few data sets on internal controls that we did with our QuantiNova related internal control RNA. Some things to consider when selecting or developing an internal control is these should be working similar or behave similar as normal transcripts. So when having different types of inhibitors, different concentrations of inhibitors affecting or delaying our CQ values of specific normal transcripts, our internal control should behave the same. And here is a data set where we did a duplex one-step RT-PCR of RNA where we did spike different amounts of, in this case, SDS into our reaction, and SDS functions as an inhibitor indeed, and we can see that the internal control with the pink bars behaves pretty similar like the various target genes we have been using here, indicated by the blue bars. So their delay, their delta CQ in the reaction is representative or is similar to normal transcripts, and this simply means an internal control can be utilized to monitor any inhibition being present in our sample. Also, here's a different set where we did 2-step RT-PCR, and in this setting here we compared the internal control to different types of genes, both housekeepers or other target genes, at the same concentration of inhibitor being spiked in the sample. And we can see that the internal control again behaves similar as the majority of transcripts, which is important because not each transcript behaves identical or really the same at the presence of inhibitors. So we need to find an internal control that over the range or over the board will always indicate if we have an issue with our sample material. Beside internal controls that will confirm the performance of our assays, there are other useful safety measures that can be included into quantitative RT-PCR to improve the precision of our results and the reproducibility. Here are particularly three other things I want to mention. One thing that is extremely useful is to integrate the removal of genomic DNA. I'm going a little bit more into details about this topic. The key thing about genomic DNA that can potentially be carried over into our messenger RNA preparation is that the GDNA also can serve as a template for our target-specific primers. And if some amount of gDNA is still present in the PCR, it will add to the signal of our specific transcript. This is particularly critical if we are looking at low abundance targets or messengers that are only creating a small signal anyway, because then an addition of a signal created by genomic DNA carryover would falsify our results potentially quite substantial. This is an issue that is not always occurring, but it can occur, particularly if we are not able to create exon-spanning primers. So the best strategy to avoid this issue is obviously to create primers that span over intron-exon borders, but this is not always possible, and if we can't design primers like that, then we always have to deal with the genomic DNA. And it's strictly recommended to remove those one as well if you're looking into things like I mentioned earlier, the MICI guidelines, for example. They strongly recommend to either look for a control against genomic DNA carryover, or much better to just remove it. And typically it's being removed by an additional DNA test that is being added to our purified RNA. That's kind of scary to add enzymes into our purified RNA that are called ASEs somehow, because they are supposed to chew up nucleic acids. They should be specific, but the procedure itself is not only an additional working step, but obviously it incurs a certain risk of something goes wrong here. And it's much nicer if the genomic DNA removal can be just integrated in the process without additional work steps. And I will show some data on that on the next slides. Another thing which is very helpful but commonly known to most people is really to use a hot start process for the enzyme, which simply means the enzyme is being kept inactive at room temperature, and only when the first PCR cycles are heating up the enzyme can start working, which avoids any type of premature enzyme activity or of primer-dimer formation, which would create non-specific noise or create a non-specific signal in cyber-based procedures. The good thing about hot start is it also allows us to work at room temperature when doing reaction setup. So it both creates convenience and higher reproducibility, but it also allows us, for example, a more relaxed handling and the option for automation, which also can add to the precision or reproducibility of our workflow. I would like to point out here that QIAGEN has introduced in our QuantiNova RT-PCR kits a so-called two-phase hot start, which for the first time really allows not only the polymerase but also the reverse transcriptase to be inactivated and only be activated at the setup of the reaction, which is very handy for one-step RT-PCR procedures. And the last point I would like to show here is a very simple tool, which is a visual pipetting, just to make sure that we can see when adding reagents to each other, there's a color change that is easily visible, and particularly when working in 96 well or 384 well setups, this comes very useful to avoid human mistakes while pipetting or taking away stress. So overall, these are integrated in-process safety measures that are taking away any type of variables or even additional handling steps to deal with them. But they are just integrated into the process. That simply means we don't have to do additional working steps here. We can just avoid this type of potential inconsistency by an integrated process in our qRT-PCR. I mentioned genomic DNA being a potential issue, and adding to a non-specific or adding to the specific transcript signal with an additional non-specific signal. And here's a data set that shows that actually genomic DNA being spiked into an RT reaction can create quite a substantial signal. The blue bar indicates a signal being created by gDNA, which is spiked into our reaction, but we have not utilized any reverse transcriptase here. So we are using an assay for detection of messenger RNA, but we are not including a reverse transcriptase in this experiment here. So there should be no signal, and this basically means the blue bars here are coming completely from genomic DNA, and the more DNA we're spiking here, obviously the signal comes earlier and earlier. And by including a genomic DNA removal, which is indicated by the pink bars, we can this signal eradicate completely. So ND means not detected anymore. That basically means the contribution or the variable being incurred by gDNA carryover can be completely removed by gDNA removal in an integrated step, which we have included in our kits. While the first slide I showed before was shown on a two-step RT-PCR where we removed the gDNA from the cDNA synthesis, this is a different data set where we have included the gDNA reduction in our one kits. This is something that is pretty tricky to achieve, but we have managed to do so, and this allows us to also run a one-step RT-PCR where we have, thanks to genomic DNA reduction, a very reproducible CT or CQ amount even after spiking genomic DNA. While without the gDNA removal indicated by the light blue bars, our CT values are coming earlier and earlier, indicating a higher amount of messenger RNA, which is actually incorrect then. Another thing I mentioned earlier is our new two-phase hot start for reverse transcription. This is a pretty nice thing because in the process for one-step RT-PCR, we are including an RT enzyme associated with a so-called RT blocker, the blue donut, which is kind of inactivated by the red dot during reaction setup. Likewise, also, of course, our PCR tag polymerase is being inactivated by an antibody and by a second component, which we call the QuantiNova guard, which really makes the inactivation extremely stringent. So at room temperature, both enzymes are inactive, and we can keep the material or the setup at room temp, and when elevating the temperature to 45 or 50 degrees, our RT blocker will dissociate from the reverse transcriptase, which in return can bind to the RNA template and create cDNA. And after successful cDNA synthesis, we're heating up to the PCR temperatures, which will both denature the reverse transcriptase and also remove the antibody and the QuantiNova guard from our tag polymerase, which then can go on and create a specific PCR reaction. This two-phase hot start is not only improving the specificity, but also allows us to do reaction setup at room temperature, because now our one-step RT-PCR can stand up to 2 hours at room temperature. This is a data set where we have looked at 3 different transcripts and either start the reaction immediately after setting it up on ice, indicated by the red lines, and the purple and blue lines indicate the same reactions being started 1 or 2 hours after the setup and leaving the whole reaction setup standing at 22 degrees Celsius, which actually means we can really improve our handling by doing automated reaction setup, which particularly in a higher throughput can increase a lot of not only precision in pipetting but also efficiency of our workflow without going the risk that anything goes wrong or our results would be altered. The next slide here, I guess it's pretty self-explanatory. I mentioned an inbuilt visual pipetting control. This is really something I personally like a lot because it's a very simple but very helpful tool to minimize any types of human errors during reaction setup. The master mix of our QuantiNova kits is blue, which already is useful when dispensing it into the PCR plates, but more useful I even find that we are utilizing or offering yellow template dilution buffer into the PCR kits, which can be utilized to do a pre-dilution of the sample material. And if we use this, then obviously our RNA samples are, or DNA samples, depending on what we're doing, these are dyed in yellow. And when adding them to the PCR master mix, we can immediately see where it already has been added, which is particularly helpful when working with 96 or 384 well format, because we can easily see which are still blue and which have turned into green, so we can just go ahead with pipetting, being ensured that we did not miss out any sample or pipetted it double into a different well. So what I would like to summarize here is there are a number of features that can be included into qPCR kits that are integrated and that ensure that we are getting highly reproducible and accurate results without any additional work steps here. These are namely internal controls, which are meant to rule out potential artifacts and particularly issues with inhibitors. This is the reduction of genomic DNA, which will improve the precision in quantification of transcripts, because we never know how much genomic DNA sometimes is being transferred into our RNA. Hot start procedures are commonly known as being useful and are now also available for reverse transcription in one-step RT-PCR. And we have also included a visual pipetting control just to make the work nicer and remove potential sources of human errors. At this point, I would like to do a little bit advertisement here and mention the QuantiNova kits that include these features. So QuantiNova kits are a complete family for either one-step or two-step RT-PCR procedures, and they include the different useful tools at those steps where it's required. Let's say if you're doing I would call it traditional two-step RT-PCR, the reverse transcription kit would be utilized on our RNA to create cDNA, and this does include an internal control that can be transcribed also into cDNA, and also it includes a genomic DNA treatment to remove any type of gDNA which can potentially interfere later on with our qPCR. And then for qPCR, then we are offering our QuantiNova either probe or cyber-based kit, which are meant for the qPCR of cDNA to detect the various transcript that you are potentially interested in, and they have the features of room temperature setup and the visual pipetting tool. And this visual pipetting tool is included actually in any of the potential high throughput procedures or reagents, which also includes our one-step RT-PCR kit, either probe-based or cyber-based for one-step RT-PCR, which also have room temperature setup ability and which also include internal control to monitor the successful amplification without inhibition here. So this is a complete product range just recently launched from QIAGEN for one-step or two-step RT-PCR. I would like to show just very briefly a few performance data on this kit. For example, when doing two-step RT-PCR, here is just one data set from our reverse transcription kit for cDNA synthesis, and this really can go from very tiny amounts like 10 picogram up to 5 micrograms, so a huge linear range where we can reliably quantify, and we can even double the protocol and then go up to 10 microgram of total RNA input if that would ever be required. So if you have huge amounts of RNA to work with, then this is possible to transcribe them totally, which rarely is the case, to be very honest. We also have created a few performance data. I'm just showing one set here with our one-step RT-PCR kits. In one step, very often, one of the critical topics is that people want to be able to detect very, very tiny amount, and we went down to 10 picogram of input material, and we can see that we are reliably able to really go down to very small amounts and still get them detected or quantified reliably, which is pretty tricky, really. We know that some other kits from other suppliers, as shown here as well in the data set when looking at the red arrows, are really running into some issues when it goes around tiny amounts, small amounts, which are difficult to detect. We can still do this, and we feel very happy about the performance of our kits and believe they are really great, wonderful kits for quantification of RNA. At this point, I would like to briefly summarize here and say controls are an indispensable part of reliable gene expression experiment or transcript quantification. Particularly the internal control helps to deal with inhibitors. Genomic DNA removal improves the precision to avoid additional artifacts. Hot start improves the setup, the handling, and avoids any variables being introduced during reaction setup. Visual pipetting helps to avoid any type of human errors, and all these components are included in our brand new QuantiNova kit family without requiring any additional purchase or work to be developed, but these controls are already made available. So the key point here is that they help to ensure that the insights, the results we're getting from gene expression research are really based on the expression profile and not on any types of variables or artifacts that can potentially be included. At this point, I would like to thank you very much for your patience and attention, staying with me, and I would like to open up for questions. So if you have any type of specific questions, please just type them into the question box. Also, additionally, we have the opportunity, you can send an email either to myself, so this is my email address here, or to our team of the webinar group, and we would definitely be happy to answer them or come back to you by email if you have specific questions. So at this point, please let me know if there is anything that we can help you or answer with. So here's one question. Are the QuantiNova RT kits compatible with TechMan type primers? Yes, definitely. So it's possible to use them both in any type of cyber detection with the cyber green kits, both for PCR of DNA, cDNA / or of RNA, and the same, of course, is possible with any type of labeled probes, not only TechMan dual labeled probes, but as well other types of commonly used probe-based detection systems are tested and it's optimized for that, yes. Another question is if the internal control has any impact on the qPCR itself. That basically is not the case. So the internal control itself is an artificial transcript that is amplified in a 200 base per length and that we have tested and also, of course, blasted against multiple species, and it's not going to interfere with any common target that you're typically interested in research or potentially interested in. So at this point, I'm not seeing any additional questions coming up here. So if you have any questions, please feel free, send them via email or let us know by any other ways. If you have other types of questions or feedback on the seminar, I would be very happy and I would highly appreciate if you let me know any type of feedback, something that you did like or something that you would miss on and would like to hear maybe in one of the future seminars or webinars that we are transmitting here, because that's the whole purpose of webinars, that we can maybe transfer some information that you would be most interested in. At this point, I would like to thank you very much for your attention. If there are no additional questions, I guess we just pull out. I would like to wish you a great day, a great pre-Christmas season actually, because that is coming soon, and a good time with friends and family. And good success in your research as well. Thank you very much and goodbye.